All right. So welcome everyone. So today we're talking about mediation, eQTLs, and then we're sort of continuing on this module of population genetics and disease. We talked about population genetics in the first lecture, disease mapping in the second lecture. Today we're talking about quantitative trait mapping and molecular traits and eQTLs, expression quantitative trait loci, and mediation analysis. And on the last lecture of the module, we're going to talk about system genomics and heritability and genome-wide measures. So the goal for today is first review last time and then build on it to look at eQTLs. So just very briefly, what we talked about last time was an intro to genetics and then how to carry out a GWAS and then how to start interpreting them. We saw these two-dimensional relationships between effect size on the y-axis and allele frequency on the x-axis. We saw that strong effect alleles that greatly impact phenotype are generally not tolerated at high frequencies. And we saw that high frequency alleles are usually only allowed to have weak effects, giving this gradient with, of course, everything populated in here and very few examples up there, but also very few discoveries down here just because of lack of power. If something is both very rare and very low effect, it's going to be very difficult to discover this. We also saw this dichotomy between Mendelian disorders that are primarily affecting protein coding genes through their strong effects, and then polygenic and complex diseases that primarily affect non-coding elements rather than genes directly. We saw the challenge of linkage disequilibrium for fine mapping. We saw that there are a large number of genetic variants associated in any one locus, out of which only one or a handful are in fact causal. And then we saw multiple strategies for fine mapping, including statistical fine mapping, but also epigenomic fine mapping based on genome-wide enrichment and then selection of variants that fall within these enriched annotations. We saw this incredible power of GWAS and how it has transformed our understanding of disease, but also the challenge of interpretation and how all of the tools that we've been learning about can actually be very helpful in mapping the non-coding genome and actually annotating these variants. We saw how the mapping of chromatin states, enhancer modules, motifs, and upstream regulators, as well as downstream target genes, can be very helpful in piecing together the circuitry of these disease loci. Instead of just saying, oh, there's something going on in this region of the genome, you can say, here are the genetic variants, here are the enhancers that they overlap, here are the cell types where these enhancers become active, here are the upstream regulators and the motifs that they recognize that are disrupted by those SNPs, and here are the target genes within these tissues, giving a circuitry of the locus rather than just, hey, here's a locus. And we saw some tools for systematically dissecting those, in particular haploreg for entering individual variants and then finding the linkage disequilibrium partners of that variant and how they're co-inherited, and all of the annotations that these overlap, including specific disruptions of regulatory motifs. And then we saw this whole concept of comparing the genetic location, the genome-wide locations of genetic variants associated with a trait with the genome-wide annotations of enhancers active in different tissues, and how we can use that to in fact map what are the likely tissues where diseases are acting through the overlap between genetic variants associated with each trait and enhancer annotations in each tissue. And then we saw how these can actually lead to sometimes big surprises. For example, Alzheimer's disease showed no overlap with brain, but instead it was very strongly enriched in these immune cells, suggesting these immune bases of Alzheimer's disease. And then how these allow us to now start clustering traits together into a network of activity. And then we saw a specific example of what it takes to dissect the circuitry of a disease locus, basically looking at this FTO region associated with obesity, how we can, number one, infer what is the tissue of action. Number two, what are the target genes, which can sometimes be far away. Number three, what are the causal nucleotides? Number four, what is the upstream regulator or regulators that are impacted by these nucleotides? And number 5 and 6, what are the cellular and the organismal consequences of disrupting these genes? And then how, by piecing together that circuitry, we can now start intervening. We can actually go and manipulate the upstream regulators, the downstream target genes, and even edit these nucleotides with dramatic consequences. For example, changing a single nucleotide out of 3.2 billion nucleotides of the human genome was able to restore the process of thermogenesis, the process of burning calories by depolarizing the mitochondrial membrane inside your beige adipocytes, by restoring the motif that is AT-rich and is lost in the risk allele carriers. So by taking primary adipocytes from risk allele carriers and editing that one nucleotide, we're able to restore that cellular process. And by changing the downstream target gene, we're able to restore the lean phenotypes and, in fact, prevent mice from gaining any weight in obesity. So that was the review. And now what we're focusing on today is the molecular traits, and specifically expression quantitative trait loci and mediation analysis. So first of all, what is the reason for understanding that circuitry of the non-coding loci and understanding their intermediate phenotypes? And then looking at some examples of both expression quantitative trait loci as well as methylation quantitative trait loci. And then diving into the methodological foundations of eQTLs, and then some insights on eQTL function and biology, and then a very powerful complementary approach of allele-specific analysis, and then lastly, Bayesian approaches for mediation, for understanding what are the mediators of the effect of genetic variance. So that basically means if a genetic variant is acting on a disease, how is it acting? What are the intermediate phenotypes through which that genetic variant effect is happening? So to recap, the motivation for eQTLs is that there are thousands of genetic loci that are each impacting the disease phenotype by a tiny little amount. And if we focus on the entire genome and the entire path at a time, that basically makes it very, very hard to discover these disease loci because these effects are infinitesimally small after the first few genome-wide significant loci. Instead, by bridging that large gap and identifying what are the genes whose expression in specific tissues is altered by these SNPs, then we can have a stronger effect. Why is that? Because there are, hundreds of genes that are contributing to the disease phenotype. And any one of those genes contributes by a small amount, but the genetic variants associated with these genes might change the gene expression by, 15% or even 20% or 50% or a factor of 3. So you might actually have relatively stronger effects on the gene expression than on the disease. Even if you increase expression by 15%, imagine if a single genetic variant altered your height by 15%. I mean, that would be enormous. But changing gene expression by 15%, that's a strong effect. So if we're able to reliably measure gene expression levels, then we can infer expression quantitative trait loci. What is an eQTL, expression quantitative trait locus? So a locus is a genetic region. And the reason we talk about L rather than SNP is because there's an association at the level of a region because of the linkage disequilibrium in these regions. So that's the locus part. And then quantitative trait basically means that I'm not going to be looking at diabetes or no diabetes, schizophrenia or no schizophrenia. Instead, I'm going to be looking at a quantitative trait. Height is a quantitative trait. Gene expression level is a quantitative trait. So when we talk about a quantitative trait locus, this is a genetic locus that controls a quantitative trait. And what is that quantitative trait? That quantitative trait is expression. So when we talk about eQTLs, we're going to be talking about expression quantitative trait loci. Let's see who's with me so far. Awesome. So 65, 35, 000. Okay. So that's at the level of expression, but that's not the only place where we can discover these QTLs. We can also look for endophenotypes. We can also look at epigenomic activity or DNA accessibility or DNA methylation or promoter activity or histomodification patterns or transcription factor binding and so on and so forth. So basically, we can define cell type and tissue specific eQTLs and any kind of molecular QTLs, such as methylation QTLs, enhancer QTLs, accessibility QTLs, and so on and so forth. And also quantitative traits that, trait loci that impact, say, the level of lipids or metabolism and so on and so forth. And that allows us to now bridge that gap between genetic variation and ultimately disease. The challenge, of course, with breaking that gap is that we have nice unidirectional arrows coming out of the genetics, because whether or not you smoke does not influence the inheritance of a particular genetic variant. So it's not that, oh, because I'm smoking, I now have a C at that location. No, it's probably that if there's a correlation between I'm smoking and I have genetic variation here, then that probably means that the genetic variation somehow influences my smoking. Similarly, if I look at genetic variation and gene expression, I know about causality. Genetic variation and enhancer activity, I know about causality. So any arrow that starts with genetics is unidirectional. But if I look at a correlation between higher enhancer activity is associated with higher expression of the nearby gene, that's just a correlation. It could be that the higher expression of the gene is in fact leading to a change in the epigenomic landscape of that region and therefore increasing that enhancer activity. Or if I find that higher enhancer activity is associated with, say, hypertension, it could be that it is a consequence of hypertension, or that it results from hypertension, or perhaps it's fighting against hypertension. So any kind of pairwise relationships that do not involve genetics, you have to recognize that the unidirectional arrows are very hard to obtain. You need to actually carry out specific statistical analysis to do that. And that's where mediation comes in. So why eQTL mapping? Why map these intermediate expression levels? The reason is that if you have a genetic variant nearby a gene, what we typically think it's doing is that it changes the expression of that gene. It doesn't impact the protein directly, but maybe it changes the level of expression of that gene, thereby altering the gene function. And of course, that's one of many different types of activities that a genetic variant might have. For example, the expression of the gene might not change, but you might change its localization, or its post-translational modifications, or its export, or its degradation, or its partners, and so on and so forth. So you can actually have many types of impact, but most of the time we're going to be focusing on the expression of those genes. And again, the motivation for that dates back to many decades ago, when Mary Claire King and A. Wilson basically saw that there are very few amino acid differences between closely related species. If you look at between human and chimp, all of these genes have almost no amino acid changes. So instead, they predicted that regulatory mutations probably account for these biological differences. And it's the same thing that we're finding when we're looking at disease studies. So if you look at, where is the genetic variation, the vast majority of genetic variation sits in non-coding regions, and more than 93% of genetic variants that are associated with disease are also in non-coding regions. So the goal of eQTL mapping is to facilitate understanding of disease susceptibility gene mapping. So how do you find the target genes of these non-coding variants? And then the idea is that you have a genetic variant that impacts the level of expression of the gene, and that indeed leads ultimately to some, disease phenotype. And when we looked at this dissection, we used eQTLs, even though, we didn't sort of belabor that point too much, in dissecting these FTO locus. So basically, when we asked what are the target genes, one of the lines of evidence for those target genes was that they were eQTLs, that the risk genotype led to increased expression of those two genes. So that's the basis of an eQTL study. You basically look at risk versus non-risk individuals, and then you ask what is the expression for risk versus non-risk individuals. And if you see an expression difference, that means that these genetic locus, in this particular case, RS1421085, and its associated nucleotides, are in fact altering the expression of IRX3 and RX5. And the reason why it helps you find the target gene is because if you look at this locus here, the genetic variants are sitting in the middle of, this FTO gene. But instead, FTO itself does not show a change in expression. The change in expression is observed only for IRX3 and IRX5 among all of these genes in this locus. IRX3, 6, no expression change. CRNDE, no expression change, and so on and so forth. Okay, so that basically tells us that the true target genes of these genetic locus are probably IRX3 and IRX5. So who's with me so far on sort of, what eQTLs give you and how you measure them and what you're actually measuring? So you're basically looking at the difference in expression between risk and non-risk individuals, or between minor allele and major allele, or between reference allele and alternate allele, for those individuals carrying that eQTL or not, that SNP or not. So 33, 50, 17, 00. Okay. So, and again, why do we care about that? Because we can then go in and manipulate these genes. And if we indeed change the SNP, we see that it has an impact on gene expression. By editing this T allele into a C, we see that it goes from low expression to high expression to low expression when I edit it back for both IRX3 and IRX5. So that establishes the SNP to gene link. And then similarly, by changing the gene through knockdown or overexpression, we can see that if I increase the expression if I decrease the expression of a gene, IRX3, upon the knockdown, you basically see that the downstream cellular consequence is in fact dramatically altered, whether I whether I knock down IRX3 and RX5, I see this increase in oxygen consumption rate. Similarly, if I overexpress IRX3 and RX5, so basically this is under control of doxycycline. Whenever I have doxycycline, I basically increase the expression of IRX3 and RX5. So that leads to this arrow of the decrease. And then here, this is under the control of siRNA, a small interfering RNA, which basically decreases the expression of that gene by degrading the mRNA. So whenever I add this siRNA, you see that it increases expression. And then this increase is higher for the risk individuals shown in red. And then they reach the level of the non-risk individuals shown in blue. So that's sort of what allows us to now paint the path of causality from SNP to gene to cellular phenotype to organism. Okay. So let's dive in. How do we study eQTLs in the context of disease? So first of all, are there any questions so far? You can add them to the chat window or you can raise your hand. But let's now dive right in and basically look at how do we discover QTLs in the context of disease. So again, as I mentioned earlier, this is looking at the relationship between genetic variation, intermediate phenotypes, and ultimately disease phenotypes. So this is a collaboration that we've had for many years now with Phil de Jager, as well as David Bennett, in the context of the Memory and Aging Project and the Religious Order Study. We've basically been looking at methylation, expression, and a bunch of other molecular phenotypes across hundreds of Alzheimer's patients and controls, and then looking at how does the methylation level of hundreds of thousands of regions in the genome vary across individuals. So you basically have hundreds of thousands of methylation probes, and then hundreds of individuals, half of whom have the particular disease phenotype. So that's the basis of a QTL study. You're basically measuring, thousands of genes or hundreds of thousands of epigenomic elements across, dozens and sometimes hundreds of individuals. So, and then the other thing that we have is, of course, a mapping of the epigenomic landscape of the same tissue, but in a small number of reference individuals rather than variation across those individuals. Is the reason you can infer causality instead of correlation just some application of Bayes' rule? It's a little more complicated, so we're going to talk about that, Ayush. But in general, the way that you find causality is through a process known as Mendelian randomization, where you basically use your genome as an indicator variable to basically effectively do a randomization study by assigning, quote unquote, SNPs to different individuals, and then using your intermediate phenotype as a sort of variable that allows you to know if indeed the genotype is impacting the disease directly or acting through that intermediate variable. And we're going to talk more about that. Okay, so here's what the data looks like. You basically have, brain with hippocampus, for example, and cingulate gyrus and anterior caudate and so on and so forth. So you basically have multiple regions of the brain that you have chromatin state annotations for, as we saw in our epigenomics lecture. And now you're going to go and see across all of these regions, which you can see are annotated as either as promoters or enhancers based on the annotations here. Across these regions, what is the level of DNA methylation across hundreds of individuals? And what is the genetic variation for the same individuals? So what's an eQTL? An eQTL is basically a correlation between the methylation vector and the genotype vector for the same individuals. So you can see here, for example, the methylation QTLs are places where you see a strong link between the two, that this spot depends on the methylation level of this spot depends on the genetic alleles that are carried at that spot. So whenever you see a line there, that's basically methylation QTL. Okay. So we basically have the chromatin state annotation, the methylation level, and the genotype. And now we can start asking, well, which regions are eQTLs? And then what are the properties of different classes of elements? For example, if I have a methylation change here, does it matter if it's a promoter or enhancer, and so on and so forth. So the first thing that you do in any kind of QTL study, in any kind of methylation QTL or expression QTL, is pre-process the data and eliminate covariates that have large effects on your data set. So the way to think about that is that there are some global patterns of variation that are not related to the local effects of the genetic variants. And it's only after correcting those that we can in fact recognize the local effects of genetic variants on gene expression. So how do you correct for those global effects? You can basically discover these by doing PCA, by doing a decomposition of your matrix into its principal components. You can eliminate the strongest principal components that are explaining a large amount of the global variation, so that what you're left with is more directly impacted by the local genetic variation. So if you look at these surrogate variables that you can discover de novo, SV1, 2, 3, 4, 5, 6, 7, you can ask how much of the variance are they explaining? And how much total variance do they explain jointly? So you can basically take the first one and then in a greedy fashion add the next one and the next one and the next one that add more and more of this variation. And then similarly, you can basically ask what are known covariates. So these surrogate variables might represent ethnicity. They might represent a global batch effect for that array that caused all of your expression levels or all of your methylation levels to be shifted to higher levels and so on and so forth. And then you can ask, how are these surrogate variables related to different types of covariates? So what you can see here is, for example, surrogate variable 1 is related to the plate. This is the 96-well plate or the 384-well plate that these experiments were carried out into. And so does SV3 and SV5 and SV6. So if you learn these covariates de novo, you can basically see that some of them relate with the batch of the experiment. Others relate with the cell mixture proportion of complex tissues. So the brain is made out of dozens of different cell types. If the sample that you have has, for example, more astrocytes, you might actually see a global shift in the methylation level related to the differential methylation of astrocytes and other cell types. Surrogate variable number 2 correlates with both age and sex of the individuals. Again, these are global effects. These are not going to be local effects. So you can basically correct for these covariates and then ask how much of the variation do they capture. So what you can see is that as you include more and more of these covariates, you capture more and more of the variation, and then eventually it flattens out. So that basically says, well, you know, we should probably include many of these initial covariates, but probably fewer of those covariates. And the way that you usually choose this threshold is to ask how many QTL target genes do I discover after correcting more and more covariates. And what you will see is that your discovery goes first up and then down. And basically first you recognize more and more of these genetic signals, and then eventually you lose that signal because these covariates are now, you know, removing these covariates actually removes signal that is part of your genetic variation. So you can now start asking, after removing these covariates, how are different chromatin states related to the DNA methylation pattern? And what you can see is that you can see what is the average methylation level of different probes. And if you look at DNA methylation, it's basically a bimodal distribution. There's many CPGs, many of these control regions that are low methylation, and these are the CPG islands that we were talking about in the HMM lecture, that are devoid of methylation, and therefore they don't lose their CPGs into TPGs by deamination of the C. Instead, they maintain them, and these are the typically promoter associated. If you look at these very highly methylated regions, these are typically repressed regions and transcribed regions. And if you look here in the middle, these intermediate methylation, these are usually more enhancer regions. So you can basically see how is this overall level of methylation related to the variation in methylation. So basically here you can see what is the level of, you know, the standard deviation of the CPG methylation. And that is actually very helpful in realizing that the highest variation is found here in the middle at these intermediate methylated regions. And then at the ends, you basically have relatively lower variation in these two modes of the distribution. And then, of course, you can ask, are these intermediate methylation levels simply an artifact of averaging bimodal distributions between individuals. And if that was the case, then we would expect to see, a high mode here and a low mode here for every one of those locations that averages here in the middle. But instead we see that they actually are, along the diagonal, suggesting that it's not just an artifact of averaging, that there's really intermediate methylation in these enhancer regions compared to, repressed regions, which are methylated, which are polycomb repressed regions, which are very broadly distributed, enhancers, which are intermediate methylated, and then transcribed regions, which are highly methylated, and promoters, which are highly unmethylated. And again, explaining this distribution here between these promoter regions that are very highly unmethylated, these transcribed regions, which are very highly methylated, and then again, all of these repressed regions, which are very highly methylated, and then these bivalent regions, which are highly unmethylated. And lastly, these enhancer regions, all of these different enhancer chromatin states are in fact showing this intermediate level of methylation. And then you can also ask, how much do the different states vary from each other? And what you can see is that enhancer regions are in fact in this intermediate regime, and they are by far the most variable chromatin state, whereas promoter regions are in fact out here in the highly unmethylated level, and then very little variation. And indeed, they are an order of magnitude less variable than enhancer regions. And again, you can see here the full distribution for every different chromatin state. Enhancers are very variable. Promoters are not variable at all. Okay, so let's see who's with me so far. Lovely, 20, 70, 10, 0. And then the last question is, how is the pace so far? Okay, cool. Most people are saying just right, just a tidbit too fast. Okay, so 27, 73, sorry, 0, 27, 73, 0, 0. Okay, so now we understand the global properties of these variation. So basically, number one, what is the high versus low distribution? And number two, what is the distribution of variability? We can now start asking, how do we discover these methylation QTLs? So here are some examples. You can basically see that we can find 50,000 of these methylation QTLs across the genome after bone photonic correction. And you see their p-values are astronomical. So for GWAS, we had p-values of 10 to minus 8, 10 to minus 9, maybe 10 to minus 20. Here we're looking at 10 to minus 300. So this is extremely, extremely significant. And the reason for that is, in part, we're doing many fewer hypotheses. So the hypothesis testing burden is dramatically reduced because we're only looking for every target probe. We're only looking at the nearby SNPs that might be controlling it. We're not looking genome-wide, number one. And then number two, because this is an intermediate phenotype, the genetic variants are allowed to have much stronger effects on these intermediate phenotypes, which are not necessarily selected against, as opposed to disease, where you have much more selection against having these strong effects. And here are some examples. So this is the typical view of a QTL. So if you have the 0 genotype, or the 1 or the 2, namely if you have homozygous reference, or homozygous alternate, or heterozygous, you basically see that the level of CpG methylation is dramatically increased. So if I'm a carrier of 2 alternate alleles, then I have a much higher level of CpG methylation than otherwise. Okay? So here's one example up here. Here's another example. Here's a third example. Here's a fourth example. So what do you notice here? What you notice is that of course, the CpG methylation level is a continuous variable, but you also notice that the SNP genotype itself is a continuous variable. whereas in fact it should be exactly 0, exactly 1, exactly 2. And the reason for that is that we're correcting for the genotype matrix as well. So we're eliminating these covariates, which basically leads to the genotype itself becoming a quantitative variable rather than the genotype always being 0, 1, or 2. So because we're decomposing that matrix of genotype by person and we're removing the principal components, the genotype itself is varying continuously. The next thing you can ask is, what is the power that I have with 25 individuals, with 50 individuals, with 100, with 150, 200, 250, 300, 400, and 600 individuals? What is the power that I have to discover QTLs? And what you notice is that, of course, the number of QTLs grows dramatically with more individuals. So as I increase the number of individuals, I increase the number of methylation QTLs that are discoverable. But if you also look at the effect size, I'm able to discover weaker and weaker effect sizes with more and more individuals. So it's just a question of power. So these methylation QTLs are increasing and increasing, but down here with 100 individuals, I can discover, QTLs that have a 40% effect size. And then out here, I can discover with 250 individuals down to 20% effect size, and out here down to 10% effect size, and so on and so forth. Okay, so that's sort of the relationship between the power that comes with larger cohorts, and the decrease in the increase in resolution, the decrease in the effect size that I can discover, and the increase in the number of SNPs. So that's the minimum effect size in red. But what you'll also notice is that the median effect size is also dramatically reduced. So the eQTLs that I'm discovering here are very strong effect, and then the eQTLs that I'm discovering here are very weak effect. The third thing you'll notice is the distance distribution. If you look at how far from their target are these SNPs sitting, you see that there's a very broad distribution of distances as you go to more and more individuals. And the reason for that is that you simply have more power to detect them. Whereas out here, you have very few and you don't have power to detect those. Okay. So then you can see here the increase in power, the decrease in the effect size. And then the other thing to ask is, in purple here, you can ask, for those QTLs that I have discovered at, say, 25 individuals, how wrong was I if I look at more individuals? So as you increase the sample size to the maximum, you can basically say, well, now I can re-estimate the effect size that I had estimated here. And you can see here this enormous error bar. That basically means that with very few individuals, your estimates of the effect size are going to be very uncertain. But even with 50 individuals and 100 individuals, that estimate of the effect size is already quite robust. And then you can see here the distance distribution between the DNA methylation probe, namely where am I measuring the DNA methylation level, and how far away am I. And what you can see is that 90% of methylation QTLs are within 75 kb, and then 50% are within 20 kb. So that basically means that even though we searched a 1 megabase interval, here you can see, that we searched just a very, very large interval. They're actually very highly concentrated close to the middle. So that's important because it can reduce your multiple hypothesis burden if you focus on a much narrower region. Of course, the challenge there is that you might miss some QTLs that are acting at a distance, but you will be able to discover many, many more QTLs because you will not be paying as high multiple hypothesis burden, and therefore you'll be able to set your thresholds to lower values. Okay. So let's see who's with me so far. So 33, 56, 11, 00. Okay, the other thing to realize is that as you increase your sample size from 25 to 50 to 100 to 600 individuals, then the minor allele frequency distribution is actually dramatically changing. So the background distribution across all individuals is that there are, many variants of weak effect, of lower allele frequencies. That basically, the further down you look, the, more in the allele frequency spectrum, the more variants you find. Basically, we carry an enormous number of very rare variants and a small number of common variants. So that's the gray bar, that's the background. But then if you ask, what is the power to discover variants of different allele frequencies, then you see that with only 25 individuals, I can only discover variants that are at 40% frequency. And the reason for that is that I have to observe them. And if I only have, say 10% frequency, I'm not going to see any people with that in a sample of 25. Basically, at most, I'm going to see 2 people carrying it, or at least on expectation, I'm going to see 2.5 people carrying it. But that doesn't give me enough power because of the noise in the experimental measurement. So that's why the minor allele frequency distribution shifts from few individuals to more individuals to basically discover more and more QTLs at lower and lower allele frequencies. Okay, everybody with me here on the relationship between allele frequency and the discovery power? So basically, when I have very few individuals, I can only discover alleles with high frequencies because otherwise they wouldn't be even noticeable in our sample. But with more individuals, I can actually discover alleles with higher, with more rare frequencies. Okay, so 22, 56, 22, 00. Okay, so, and then now you can ask not just what are the chromatin state positions of the probes themselves, which is what we did earlier, but now we can ask where are the methylation QTLs sitting? And what you can see is that there's very few methylation QTLs, there's very few SNPs, there's very few genetic variants that control methylation in promoters. By contrast, there's a lot of variants that control methylation in enhancers. So that's kind of interesting. It basically tells you that perhaps individual genetic variants don't have as much of an effect in a promoter region because there's so many other things that are converging onto that promoter. But in an enhancer region, which is much more distant from the gene, then a single SNP, and therefore the binding of a single regulator, might actually have more of a genetic effect. Whereas here, any one regulator probably has little effect because there's just so many different points of convergence with many different enhancers contributing, and so on and so forth. And then lastly, you can ask, what is the variation in methylation and how does that relate with the specific disease diagnosis? And in this particular case, if you look at the ability to classify Alzheimer's versus non-Alzheimer's individuals based on the level of methylation in a region, you see that those probes that sit in enhancers are much more predictive of Alzheimer's, and those probes that sit in promoters are much less predictive of Alzheimer's. So that basically means that genetic variation, that not only are enhancers more variable, not only are enhancers more genetically controlled, but the genetically controlled enhancers are more likely to be having an impact on disease variation. So basically the prediction power of Alzheimer's is much higher if I look at methylation in enhancers rather than in promoters, whether or not it's genetically driven. And then what's really remarkable is that these enrichments for specific chromatin states persist across thousands of probes. So basically enhancers are, overrepresented for, thousands of probes, suggesting again these thousands of weak effect loci all contributing to these disease phenotypes. So what we can actually start doing now is looking at the relationship between genetic variation and disease in the context of these intermediate phenotypes. So a genome-wide association study is the relationship directly between genetic variation and disease. And for that, you need tens of thousands of individuals because you have very modest effect sizes, and therefore you need a large power. If you look at methylation QTLs or expression QTLs, you basically need, relatively fewer individuals, only on the order of hundreds, to basically learn the relationship between genetic variation and these intermediate phenotypes. And that's, again, a unidirectional arrow. A methylome-wide association study, kind of like a genome-wide association study, instead of looking at genetics to disease, is looking at methylation to disease. So that's an MWAS, kind of like a GWAS. So an MWAS is looking at the correlation between DNA methylation and disease. But notice this arrow is bidirectional. And the reason for that is that the methylation differences might simply be a consequence of the disease. Everybody with me so far? Yep. So by contrast, if I look at the first genetic relationship here between genetics and methylation, if I'm able to predict the level of methylation that a person will have when they're 93 years old in their brain at birth based on the genetic variants that they have, maybe I can actually use predicted methylation rather than observed methylation. And that's where it gets kind of interesting. If I look at imputed methylation, which is the genetically driven methylation where I apply this G2M to predict imputed M, then I can ask, how is imputed methylation correlated with disease? And because imputed methylation is genetically driven, this is not all of methylation, this is only the genetic component of methylation, then I have a unidirectional arrow again. And one of the things that you could do is basically look at what is the discrepancy between this bidirectional arrow and this unidirectional arrow in order to find DNA methylation regions that are influenced by the disease, where the arrow is in fact flowing the other way rather than this way. So here, the key idea is that we're going to learn a G2M model. This is predicting methylation from genetics across hundreds of individuals. And then we're going to impute methylation across the full cohort of individuals. So we are not constrained to only look at the 800 individuals that we could do in MWAS. Instead, in an imputed MWAS, you can actually carry out this association for everyone you have genetic variation for, without actually necessarily having to measure their methylation in their brain. Okay. And when you do that, so first of all, well, who's with me so far on this slide? Basically, the concept that I can learn a G2M function, apply it to all of the Gs to predict M, and then correlate imputed methylation with disease across the much larger cohort. Okay, so 33, 44, 11, 11, 0. So when you do that, you basically see that even in locations that didn't have any genome-wide significant hits, so you can see here that the GWAS score is, not genome-wide significant, because it would have to be 5 times 10 minus 8, which is going to be up here above the 7. You find, however, multiple locations that are now genome-wide significant based on their imputed methylation score. So you're going to impute methylation using not just one SNP, but combinations of SNPs. And then look at the correlation between that imputed methylation level and disease. And that allows you to now start finding genome-wide significant loci, which are above the dashed line here, which would not meet genome-wide significance threshold here. Only one of them is actually very suggestive, but several others are just further down. And the reason is that we are using multiple SNPs together to predict this rather than any one SNP in isolation, which simply lacks power at the genome-wide level. All right, so what we've talked about is number one, the motivations for mapping regulatory variation, and number two, a specific case study of methylation QTLs in the context of Alzheimer's. So now let's look at the conceptual framework of eQTLs and then specific methodologies on eQTLs. So what is the conceptual framework for mapping these regulatory variants? Basically, we're looking at gene expression as a quantitative trait. So the key idea is, I'm looking at genetic variation and how it might impact initiation or transcription or translation or degradation and so on and so forth. And then for cis-acting variants, what I'm going to look for is the relationship between genetic variation in that location and the expression of the gene nearby. For trans-acting variants, I might actually have genetic variation that impacts the regulator. For example, it disrupts the DNA binding domain of that transcription factor, and then it can no longer bind across all of its targets. So this would be a trans-acting relationship. And then, of course, you can have many additional levels of regulation after that. So everybody clear as to the cis-acting is looking at the effect of nearby variants, and then the trans-acting, kind of like transcription factor, the trans means from across, kind of like transfer, that means carry across. So trans-acting means from somewhere else in the genome, whereas cis-acting, cis means on the same side. So cis-acting means that this is from this location of the genome where this genetic variant impacts the gene nearby in cis, as opposed to a regulator far away in trans. Okay, so let's see who's with me so far. Lovely. So 30, 60, 0, 10, 0. Okay. So basically, if you look at gene expression as a trait, that's where the quantitative trait part of the eQTL comes in. You can basically ask how heritable are different transcripts. And you can see that well-annotated transcripts from RefSeq are very heritable. So that, and they're much more than what you would expect in a simulation. So that suggests that gene expression actually makes sense as a quantitative trait. And there's many different types of variants that could affect gene expression as a quantitative trait. You might affect TF binding, you might affect DNA methylation, you might affect histone modifications or insulation, and you might affect splicing, and you might affect post-transcriptional degradation through microRNAs or through other mechanisms. And then the cis variation is usually through the transcription factor that binds a motif that might now be disrupted in a promoter, in a splice site, in methylation, or in chromatin modification, whereas a trans-acting eQTL is usually a genetic variant that changes either the expression of that gene or that changes the binding affinity of that regulator for the particular target. All right, so now let's turn to what are the methodologies that we can use to, in fact, discover eQTLs. So we're going to use a regression framework, but also other frameworks. So here's the basic concept of an eQTL. You're doing two things. On one hand, you're measuring the loci themselves, the genetic variation through DNA, and you do this through a genotyping array across millions of SNPs, followed by QC, etc. That's the genotype part. And then you also measure the expression. That's the phenotype part. The expression by isolating RNA and then measuring the expression across hundreds of genes and hundreds of subjects. And what you end up looking for is a linear regression relationship between the output variable, which is your phenotype, this is the expression level, as a function of some baseline beta, and then some effect size factor, that's the beta 1, that's the scaling factor, that's the effect size of that genetic variant on the expression, plus some error term. Okay, So you're distinguishing between some baseline expression, beta 0, and then the variation of that in the slope for carriers of homozygous alternate versus homozygous reference. Okay. Who's with me so far on this sort of very basic regression equation of carrying out a QTL analysis? So we're going to be looking for places where this beta is non-zero. So roughly 50-50, 0, 0, 0. Okay, so what we're going to be doing is basically looking for this linear regression where we can express expression level as a function of genotype and the covariates. And what are these covariates? These are the same things we talked about earlier: age and sex and population, ethnic background, plate, bisulfite levels, and other kinds of technical covariates. And we're going to be asking how much of the expression can I predict as a function of genotype plus covariates. And then we're going to be looking at every single SNP in this QQ plot to look for what is the right significance threshold where the calibration is very good, and then it starts deviating from that. And then we can annotate them, we can visualize them, we can interpret them, and so on and so forth. So there's many considerations. First of all, how do you measure expression? You can use a microarray, or you can use RNA sequencing. How do you pre-process? How do you normalize? How do you select specific features? Do you only use uniquely mappable elements? Do you look for SNPs that might be disrupting the probes themselves in your microarray, and therefore, that array will no longer be binding in the first place? And these are sort of technical considerations and all kinds of biases. And then there's also statistical considerations about power, namely, what is the minimum expression level that you're going to be looking at? What is the minimum population variance that you're going to be looking at? So to get a little bit more into the SNP under probe effect, you basically can have SNPs that directly impact the hybridization between your measurement and your the values that you're inferring. So if the SNP is actually directly perturbing the measurement, you can't really call it an eQTL. You would have to sort of measure the expression somewhere nearby without a SNP. So that's sort of why the SNP under probe is something that you want to eliminate. So basically, if it's at the center of the probe, it might very strongly influence hybridization. But if you have multiple probes together, then of course they're less influenced by a single variant. The second thing to ask is, what is your overall variability in the gene expression? Namely, what you're trying to figure out, as I was mentioning in a previous lecture, I believe the last lecture, is the difference between the risk allele carriers and the non-risk allele carriers. And if 0 is here, and these guys are at 1 and these guys are at 4, then you could say, okay, great, it's a 4-fold effect size, right? But it actually makes a very big difference if it's a 4-fold effect size with a very broad distribution, or if it's a 4-fold effect size with a very narrow distribution. And the variance in your intensity is actually going to have a huge effect on your ability to distinguish the actual effect of that SNP. And the reason for that is that in your measurements, all of your points are going to be here, very tightly distributed. And then all of your points there are going to be here, also very tightly distributed. But if instead you have a very large variance, then your points are going to be all over the place for one distribution. And they're also going to be all over the place for another distribution. And you see here that many of the orange and the red points are in fact overlapping. So it's going to be very hard to sort of say, oh, this is caused by the genetic variant, because the two distributions greatly overlap. Okay. So who's with me so far on the impact of variance on being able to actually discover these eQTLs? So the higher the variance, the less you will be able to tell whether something is an eQTL or not. So 40, 60, 0, 0, 0. Okay. So you can also ask, how am I defining the minor allele frequency? Is there a threshold that I'm using? How do I define the search space? Do I only look at cis acting variants? Do I look at all of the SNPs, which is truly genome-wide? What is my search radius, basically surrounding this probe that I'm looking for? Do I look at 1 megabase on either side? Do I only look at 100 kb, as we saw in the previous slide? And other considerations are, how do I adjust for my covariates? Do I use demographic covariates like age and gender? Do I use population stratification to understand the structure of the underlying population and use specific controls? How do I control for batch effects and for unobserved confounders? So ethnicity is influencing both your expression levels as well as your SNP. So you don't really want to find tons of SNPs that you can simply say, well, every person of Asian background has higher expression, and therefore every single SNP in the genome that is associated with an Asian ancestry is in fact associated with that QTL. I mean, that will be a lot of false positives because they're not acting at the cis level. They're not really controlling things genetically. So that's sort of the challenge that you need to do there in controlling for these ethnic specific differences. So you can estimate this population stratification, and we saw this map of Europe earlier, where you basically have Russia here, the UK here, you have Spain down here, and then Sweden up there, and so on and so forth, which correlates very much with the axis of migration in the European continent. So you can basically summarize not just your expression levels and remove expression-associated PCs, but you can also remove principal components of your genetic factors. So you basically have your expression as a function of your genotype, and of course, as a function of your principal components of overall genetic variation, plus all kinds of other covariates. So basically, after you correct by these expression PCs, you might realize that, wait a minute, all of the samples that have that were profiled on that date are in fact falling much further down along the first principal component than the samples profiled on a different date. So that suggested that there's a strong batch effect, and that strong batch effect is in fact principal component number 1. This is actually explaining a huge chunk of your variation. And if you're not careful, then you're losing a lot of your signal because of these variants that we talked about earlier. Namely, if I have contributors to variation that I'm not measuring, these contributors are going to be shifting my samples up and down more or less randomly because they're not going to be dependent on that one variable, which is whether I have a G or an A. So instead, I'm going to have very broad distributions. So by narrowing down the distributions, by eliminating these confounders, I can basically have tighter distributions and therefore more power. So any kind of variance that is undesirable can basically reduce your power on this. So we can basically adjust for the expression principal components, and what you can do is measure as you include more, as you're correcting for more and more principal components, what is the total number of eQTLs that you're discovering. And what you can see is that after you include 10 principal components, your power is maximized. You discover many more QTLs. But after that, your power is reduced again. And the reason is that here, in the first part, you have a lot of covariates that are not, driving the biology. And after that, and therefore correcting them will yield a cleaner signal. But after that, you're effectively correcting for part of your signal as well by removing additional covariates. Okay, so let's see who's with me so far. Lovely. So 33, 66, 000. Okay, so now instead of just looking at the traditional regression framework that we saw earlier, where your expression depends on your genotype with some beta, instead what we're going to be doing is asking, well, my expression depends on some baseline alpha, and then my genotype at that location, so all of the nearby SNPs, and instead of just plus error, plus some effect of gender, some effect of age, some effect of each of my genetic principal components, and some effect of each of my experimental principal components, plus some error. Okay, so everybody with me here? So, I mean, this is the, the basic framework of an eQTL analysis. So it's a regression analysis where you're asking for that beta, and you're doing a test of whether that beta is non-zero, and what is that beta? And then you're looking for not just one beta with a local genotype, but also all of the other covariates, all of your genotype, de novo learned PCs, and all of your expression de novo learned PCs. Okay, so 44, 56, 000. Okay, so as you're carrying out your eQTL studies, you would like to know whether you should apply a cutoff of your minor allele frequency. Should I only look at variants that are greater than 10%? Am I underpowered? Maybe I should only be looking at those. What kind of normalization should I use? Should I use support vectors, principal components, and so on and so forth? How far should I search? Should I only search 25 KB to 150 KB, 2 megabases? And then what kind of filtering should I use for those genes, and so on and so forth? So depending on each of those decisions, you can ask, how many genes do I find that have an eQTL controlling them? So deciding on, whether you use an FDR of 0.01 or 0.05, you can basically see this, there's many more false positives here, but of course, many more discoveries as well. That's number one. But then, regardless of what FDR you're using, you can ask, what is my minor allele frequency? If I include more and more variants by going to lower and lower allele frequencies, do I discover more SNPs, more genes with eQTLs? And the answer is that yes, sure. As you include more variants at low frequency, then you discover more eQTLs, but up to a point, and then it drops off. And the reason it drops off is because of multiple hypothesis testing. You simply don't have the power to discover more of those QTLs, but you're testing, your multiple hypothesis burden is increasing dramatically, causing the total number of genes at a given FDR to drop. Is everybody with me here and sort of what we're looking at here? So what we're basically asking is, how is the threshold that I'm using for the minor allele frequency influencing my overall power? And we're measuring that power based on the number of genes that have an eQTL. Awesome. So 44, 56, 000. That's, well, one type of influence. The other influence is, what is the radius that I'm searching? If I search only 5,000 KB, then I, have a certain power. If I search more, it decreases, it increases. And then if I search a very large radius of 75,000 nucleotides, then you can see how my power is actually decreasing dramatically. And the reason for that is that I, again, have more multiple hypothesis tests, and I have more false positives. And therefore, when I correct for my FDR, that is actually going to drop further. Okay. So that basically tells you that going further than 75,000 to like, a million nucleotides, will reduce your power dramatically. So the challenge also with rare variants is that there's a lot of statistical variance that basically, as I showed you earlier, the uncertainty with which I can estimate the effect of that eQTL increases greatly when I have lower effect sizes. So I see 2 comments in the chat. Oh, yeah, I cut out for a moment. It was saying that my internet is not stable. Am I back? Can you guys hear me again? Yeah, awesome. And then, of course, the number of principal components influences things greatly, and so on and so forth. So you can basically ask, how do I select these parameters? What is the number of genes that I'm discovering as I include more principal components or as I change my minor allele frequency? And how do I move down that space? Of course, each time correcting for the multiple hypotheses stemming from the meta parameters, the sort of hyperparameter search as well. So then we can tune this search accordingly, and then have more power. So Ben Raby has basically developed this bioconductor package for carrying out these eQTL analysis, GG tools, that allows you to modify all those parameters. And I really encourage you to use it when you're looking for QTLs. So that's basically the basic framework for discovering eQTLs. We can now start talking a lot about what have we learned about eQTLs and their biology. So what have we learned about eQTL prevalence and impact? We're basically learning that eQTLs are everywhere, that basically with increasing power, you find more and more eQTLs across many different tissues, that they're positioned usually very close to the transcription start site. There's a 34-fold enrichment within 5 kb of the TSS. We can learn about reproducibility across different data sets. And then that is, of course, a function of power. And you can look at how tissue specific they are. And then GTEx, this genotype tissue expression project, has basically looked a lot at the reproducibility of eQTLs across different tissues and how consistent they are. And what we're finding is that the sample size is a great factor influencing the number of E genes, and that as you increase the sample size, you're just discovering more and more genes. So nearly every gene has some genetic control in very different tissues. You can also see how these cluster between different tissues and how much sharing there is in these eQTLs across tissue. You can see here a big block of sharing between different regions of the brain, and also just other blocks between muscle and heart, and so on and so forth. You can also ask, what is the annotation of the nearby genes based on the QTLs that you're finding and the biological functions that the genes carry out. And then what you're finding is, in some cases, the same SNP can actually have multiple co-regulated target genes in the same neighborhood. And in terms of transacting QTLs, one of the things to realize is that the transcription factors that target multiple genes will basically be trans QTLs for dozens of genes rather than just trans QTLs for one location. So you can use that across the genome to basically look for significant QTLs. You can also see that by including QTLs in your studies, you are able to predict GWAS loci with higher and higher power. So by filtering only by the distance from the gene or the minor allele frequency or the chromatin state, you get to more or less power. And you can see here, for example, that focusing on enhancers can increase your power and these sort of setting. But looking specifically within eQTLs that are significant can dramatically increase your power for GWAS variant discovery. So that suggests that eQTLs are in fact very relevant when you're searching for GWAS SNPs. So if you search without regard to eQTLs, then you only have a certain power. But if you search for GWAS variants within eQTLs, then your power basically continues to increase, and you can discover many more eQTLs that way, many more GWAS variants that way. I also want to spend a little bit of time talking about an orthogonal type of analysis to eQTLs. So that's allelic information. So what are eQTLs based on? So basically, if an individual has two A alleles at a location or two G alleles at a location, then you can see the difference between those individuals carrying GG or those individuals carrying AA at any one particular location. And of course, the heterozygous individuals will be somewhere in the middle. But the question is, why are the heterozygous individuals in the middle? One reason might be that their G allele is in fact behaving just like the GG here, and their A allele is in fact behaving just like the AA here. And if I measure overall expression in these heterozygous individuals, then I might not be able to see that difference between the G allele and the A allele. What allele-specific analysis does is it specifically asks for those reads that are produced by the chromosome of that individual carrying the A allele, are they different from the number of reads produced by the chromosome of that individual carrying the G allele? So you're looking in allelic analysis, you're looking specifically within heterozygous individuals and at the difference between the two alleles of that person. So who's with me here on the allele-specific analysis? So whereas QTLs are just looking at across individuals and comparing the reads from one individual, from another individual, from another individual as a single bucket, what allele-specific analysis does is it splits the reads of one individual into the major versus the minor allele, or the reference versus the alternate allele, and then specifically looking for differences within each cell of that person between the G and the A allele. So 30, 70, 0, 0, 0. So you can also look for allele-specific eQTLs, and then the concept is the following: if I have the G genotype or the CC genotype, then what is my allelic imbalance for a nearby gene? So I can basically ask, what is the allelic imbalance between the reads that are produced from the T or from the G variant in this location versus what is the allele setting in some nearby region. Why am I doing this? The reason is that our individuals are not always haplotyped. So it's very difficult to infer whether that individual carries the G and the T or the G and the G at that location. And therefore, if all I have is expression levels here for each allele and let's say accessibility level here for that allele, it's very difficult to know whether the individuals that carry the G are in fact expressing the T or whether the individuals carrying the G are in fact expressing the other G. But what you can do instead is ask for an allelic imbalance in the heterozygous nearby locations. And if you find that the allelic imbalance is higher for the heterozygotes, then that suggests that that SNP is probably controlling the expression of that gene. Okay, so you're basically testing for these two hypotheses of whether the haplotype imbalance ratio distribution is the same, no matter the eQTL SNP, or whether the imbalance is higher when the eQTL SNP is heterozygous compared to homozygous. Who's with me here on this slightly more subtle point? So because I don't have linking information, because I don't have haplotype level information, I basically have to resort to looking for whether that eQTL, whenever heterozygous, is actually leading to an allelic imbalance of the nearby locus. All right, so 0, 75, 25, 0, 0. Okay, so, but you can also combine the two types of information. You can basically look at allelic plus eQTL information. So in the heterozygous individuals, you can look for allelic imbalance. And in the homozygous individuals, you can look at the overall read depth. And you can basically have both the allelic imbalance component using, for example, a beta binomial distribution based on your input parameters, or the total read depth for the QTL information using a beta negative binomial. And then using the two together allows you to now increase further your power to discover, for example, GWAS replicated SNPs. You should also think about epi-alleles. These are nearby epigenomic signals that influence allelic activity. So you might realize that in the context of methylation nearby, that SNP might no longer exert the same effect. So these are known as epi-alleles, which are basically, depending on whether you are methylated or unmethylated, then you might actually have the ability to observe allelic effect or not. And then lastly, in carrying out these mediation analysis that I mentioned earlier, you can basically now start asking whether the specific SNP that you're observing is having an effect on expression. And the way to do that is you could, as I described earlier, impute the expression for every person in a genotype cohort. So you could basically say, well, I have my SNPs, I have my relationship here between genetics and methylation, so I can just impute methylation. So that's one approach where you're observing the SNPs and then predicting methylation and then correlating that with the trait. But an alternate approach that was pioneered by Sasha Gusev and others is summary-based transcriptome-wide association study. And the idea is the following. It's a subtle idea, but it's a very powerful one. You can use the summary statistics directly to get to the associations. And the reason for that is if I predict expression, I'm basically collapsing the uncertainty of my signal. I'm sort of using a maximum likelihood estimate as opposed to capturing the true variation of my signal. But if I instead realize that, wait a minute, it's a linear relationship between these SNPs and the expression. And the same linear relationship that allows me to predict expression using multiple SNPs at one location, and then predict the relationship with the trait from the predicted expression. Because I have two linear relationships, I can just simply combine them and then directly predict the effect on the gene by using the LD scores without necessarily needing the individual level genotypes. Because of this linearity of these relationships, instead of basically saying, oh, I need to have the genotype of each person to predict their expression, well, that's a nuisance parameter. I might not need it at all. Instead, I can just infer the global relationship through expression for that trait by combining simply the summary statistics for every single one of those SNPs. And if I use the summary statistics of those SNPs, I don't need to actually resolve this. I can just simply say, well, this SNP is acting that much on the expression, and that SNP is acting this much on the expression. And I can take the combined effect of all those SNPs at the level of the summary statistics for that individual, and then infer the effect of the transcriptome on this. So you can basically use a Bayesian linear regression model for modeling these eQTLs, for basically asking, what are my observed factors? What are my hidden factors? And how are the SNP eQTLs affecting the expression levels directly? And then you can, of course, add a certain amount of noise. So these are the covariates, for example, which are both observed and inferred. And these are the SNPs and then the eQTL effects here. So you can extend these or linear regression models using this Bayesian framework. And the concept is that instead of just asking what is the maximum likelihood estimate for my parameter, I can actually use a prior for my parameter to select relevant variables. I can use different random effects models. I can actually I can actually sample the linear mixed effect in a sparse kind of way. And I can also fine map causal variance using the LD correlation. So for the spike slab prior, I can basically say, well, I can use a Laplace prior, or I can use a spike slab prior, which basically is a combination of a spike exactly at the value, and then a slab that avoids having zero probabilities and infinite penalties in the context of these distributions. And then I can search for the optimal parameters that allow me to now fit the model within this maximum likelihood and maximum a posteriori framework. You can use these random effects in the context of your population structure to basically study the kinship matrix relationship and then infer that and remove it. And you can also use these random effect models to basically look between an uncorrelated or a reference corrected null that basically better follows the true distribution. And I really encourage you to look at the work by Jennifer Listgarten and others on linear mixed models and how they can be used for actually correcting for a lot of those effects, for calibrating the null distribution and then capturing significant variables without this overinflation that can stem from population structure and other effects. So you can also use these sparse linear mixed models by basically forcing many of your variables to actually become zero by using these, two mixture model, one mixture with a causal effect and this other mixture with an infinitesimal background effect that basically pushes many of these variables to become zero. And you can also fine map causal variance using this approach by sort of selecting out those variants through the LD relationships that they have and the expectations of those, that those LD relationships impose on the p-values that result. So you can basically do this fine mapping under the hood by basically using these summary z-scores, an unknown genotype and an unknown phenotype vector, and then assuming that the phenotype vector was generated according to this distribution, which basically should follow this, and then considering the potential collinearity embedded in this R matrix, and then using this spike slab prior. And for computational efficiency, you can basically restrict the number of causal variants to, for example, not more than 3. So there's different types of estimation algorithms that you can use. You can use exact inference, you can use Markov chain Monte Carlo, or you can use variational Bayes. And then there's many different ways of doing this, and I really encourage you to look up these methodologies. So what we talked about today is the motivation for mapping regulatory variance, the overall conceptual frameworks of eQTLs, and then different methodologies for eQTL discovery, then some insights on eQTL function and biology, and then this complementary approach of allele-specific analysis, and then lastly, Bayesian methods for carrying out mediation and TWAS. So that's where I'll stop for today. Last question, who feels that they've learned something? Lovely. Okay, so yeah, it's 33, 67, 0, 0, 0. This is lovely. Okay, thanks everyone. See you at the mentoring session tomorrow. So we'll treat it as office hours, and you guys can come and discuss your end-to-end pipeline results. Talk to you tomorrow. Bye, guys.